which is basically differences. This says give 1 point difference between the following pairs as indicated in the brackets. Now please note, whenever you're working on your differences, you have to draw it in the form of a table. Otherwise you don't get any marks. So put down your table, use a ruler and a pencil, make it very presentable, and put down your hypertonic on one side. This of course is your first question, and hypotonic solution on the other side. Now make sure you use a ruler and draw a nice straight line down. Okay, and this says difference between these two based on their definition. So what then is a hypertonic solution? Now that basically is a solution which has a lot of solute in it. Yeah, so we could put this down as a solution that has a lower concentration of water water molecules than the cell sap. Whereas this is a solution that has a higher concentration of water again compared to the cell sap. Now always look at your hypertonic and hypotonic solution in terms of their concentration of water molecules. In terms of hypertonic solution, it has a lower concentration of water molecules, whereas hypotonic has a higher concentration. Now make sure your table is nice and neat and complete with the help of a ruler. Make it very presentable. That's your first one done. Let's move on to the next one. Myopia and hypermetropia, where we're going to be talking about image formation site. So we have the second one, myopia and hypermetropia, which may also be called hyperopia. You've got a table there. Now, image formation site. Now, work this out in your head. Myopia is short-sightedness, which means in myopia, a person is able to see close by objects. But when the person looks at far-off objects, which we depict like this, the image falls short of the retina. Whereas in the case of long-sightedness, this person can see far-off objects, but when this person looks at close by objects, the image falls beyond the retina. So there you have your answer. Image is formed in front of the retina and in this one the image is formed behind or beyond the retina. Complete your table neatly. Excuse my lines because I can't use a ruler over here actually. But when you do it, make sure it's nice and presentable. And the third one we have is the sympathetic nervous system. Now note how I'm using block letters for these headings. Although you don't have to do it. I'm using this to differentiate my heading from the points that I'm going to be writing. and this is number 3. Don't forget to number it neatly. Okay, so your sympathetic nervous system, the function. If you remember, sympathetic nervous system works on your involuntary activities and is triggered off by adrenaline. So you could put down any of the functions that are there, such as your heartbeat speeds up, and prepares the body to meet an emergency situation. Here we can put down heartbeat slows down and helps the body to return to its normal state. Once the crisis has passed. Now you could have just simply written down that this prepares the body to meet an emergency situation as well, and this one just helps the body to return to its normal. We've taken an example of heartbeat speeding up, but we've got several others that we had discussed as well. Okay, so this done, we now move on to the fourth one, which is the difference between osmosis and diffusion. We're supposed to differentiate these based on the explanation of their terms. This one's simple enough. Osmosis is basically the movement of water molecules from the region of their higher concentration to the region of their lower concentration across a semi-permeable membrane. There is a temptation to write some words such as concentration and membrane in short. Avoid any such temptation and make sure you put down the full form of it. Okay. Diffusion, on the other hand, is the movement of molecules of a substance this could be a solid, liquid, or a gas from the region of their higher concentration to the region of their lower concentration when the two are in direct contact. Is that clear? Now these two definitions are very important. Either ways, you should be knowing them really well. Okay, now moving on to the next one. That is the rod and cone cells of the eye. So we have the fifth one, the rod cells. On this side, the cone cells. In terms of the sensitivity towards light. So you're supposed to know that the rods are very sensitive to light. And can perceive even lesser intensities of light. The cone cells, on the other hand, are less sensitive to light and can only perceive moderate intensities of light. Right, this done. Let's move on to the next question now.